Good morning, everyone. Welcome to the May session of the NCAS seminar series on this very fresh Wednesday morning. At least it's nice and sunny. I'm Ngan-Ha. I'm a pharmacist and project officer at NCAS and your host today. So before I start, I'd like to acknowledge the traditional owners of the lands on which we present today and pay my respects to elders past, present and emerging. So today we have a very interesting focus on the microbiome, and we have two wonderful presenters, Dr. Olivia Smibert and Mr. Tony Lai, who will be talking about the unseen antibiotics, effects of antibiotics, and also phage therapy. So our first presenter is Dr. Olivia Smibert, and she is the lead for Transplant Infectious Diseases at Austin Health and also doing her PhD at Peter Mac and the University of Melbourne. Her PhD project aims to identify the role of the microbiome in determining infectious and immunological outcomes in immune-compromised specialty patient populations, and then how this could lead to novel therapeutics and diagnostics. So I'm so excited to hear Olivia's presentation today. And so over to you, Olivia.
Thank you very much. Hopefully, am I sharing my slides now that all looks okay?
Great.
Perfect. So thank you for the kind introduction and for the opportunity to speak to you today. I'm going to be chatting about antibiotics, the gut microbiome, and the effects that we may not see. And I probably should preface this by saying I'm really going to be focusing on anaerobic antibiotics, or this is what I hope everyone will take home, anaerobic antibiotics, the anaerobic microbiome, and the effects that we may not see. So I can't really start this talk without sort of going over exactly what the microbiome is and why it's important to us. So our bodies are host to trillions of microorganisms that we collectively refer to as the microbiome. They vastly outnumber us. There are 10 times more bugs than there are human cells on us. So if we decided to go left and our bugs wanted to go right, we would be going right. And the greatest density of these bugs are found in our gut, and they're mostly composed of strict anaerobes, which really means that they do not require oxygen for life or metabolism. In a healthy state, our microbiome performs a number of functions that are essential to life. They help us recover energy from the metabolism of non-digestible food components. They help prevent colonization by organisms that would be pathogenic to us through production of antimicrobial compounds and through outcompeting the presence of resistant organisms in our gut mucosa. And they really importantly modulate our immune system through interactions at the gut wall. So this reciprocal relationship between the microbiome and our immune system really is essential to life and determines whether we're in a state of health and wellness. And disruption of our gut microbiome is associated with a plethora of human diseases. And just about every human disease you can think of has been linked with a disrupted microbiome. And obviously this is a focus of considerable research effort, because it may well be that modifying or optimizing our microbiome or targeting our therapies to the microbiome may have untold effects in a multitude of disease settings. So antibiotics also are essential to life, unfortunately. Over half of hospitalized patients receive at least one antibiotic. We know they're critical at saving lives, particularly in the setting of sepsis. However, while they are wonderful, there are also some untoward effects. And unfortunately, it's estimated, depending on where you look, that a considerable amount of antibiotic use may be inappropriate. What do we know about the impact of antibiotics on the microbiome? Well, we do actually have a lot of literature to tell us from preclinical and clinical models exactly how damaging antibiotics are to our gut bugs. So looking at animal models, exposure to antibiotics has consistently been shown to cause loss of our healthy anaerobic gut bugs, domination with potential pathogenic gut bugs, translocation of gut bugs, so the allowance of gut bugs to pass through the bowel wall and circulate in the bloodstream, and then that leading to markers of increased systemic inflammation. And the point I want to make here is that the effect of antibiotics on our gut bugs, which we think may be limited simply to the lumen of the gut, can actually be associated with far-reaching effects and immune responses distant from the gut or outside the bowel wall. In particular, broad anaerobic spectrum antibiotics are particularly damaging. And I think the most concerning mouse study I've ever read was undertaken by Shono et al. Group, who were looking at the effects of gut bugs and how they influence graft versus host disease in a mouse model of stem cell transplant. When they tested a variety of antibiotics and the effects that this had on the gut microbiome of mice, they found that just 48 hours of piptazo essentially eradicated the microbiome entirely from the bowel of a mouse, with no DNA from a single bacteria able to be amplified, which is pretty concerning. So what do we know from human challenge studies? Well, we've seen similar things, to be honest. If you expose healthy adults to antibiotics, you find that there is a loss of diversity in the community of gut bugs. There is a significant change in the composition of your microbiome communities, and in particular, loss of these healthy anaerobic gut bacteria, particularly those that elaborate what we call short-chain fatty acids or butyrate, which are chemical compounds that are integral for interacting with our bowel wall and interacting with the immune system that's resident in our bowel wall. We also see increase in the abundance of antimicrobial resistance genes. So the effect of antibiotics, they happen quickly. They occur within days of exposure, and this is being consistently demonstrated. And the microbiome of our gut, once we've been exposed to antibiotics, actually takes months to recover. And in some instances, there is permanent disruption of the bacterial community, months to years after exposure. And the most damaging effect, again, as we saw in animals, are these broad spectrum anaerobic antibiotics, presumably because they disrupt our health-associated anaerobic bacteria. So what do we know about antibiotics as an independent predictor of non-C. diff and non-MDRO clinical outcomes? And sorry, my screen dropped out before. I think probably my key point to summarize all of this together is we know that our healthy gut microbiome is important. We know that overuse of antibiotics and exposure to antibiotics leads to disruption of the gut microbiome and can cause subsequent downstream disease, with the most well known to us being C. diff, but also colonization with multi-drug resistant bugs and invasive infection with multi-drug resistant bugs. So I think it sort of sets a setting that perhaps there are more untoward effects of antibiotics that have been poorly characterized because we don't know exactly all the different ways that our healthy gut bugs are essential for health. So that sort of leads me to the next section, where I really want to look at healthy adult populations or general adult populations seeking healthcare, and what are the untoward effects of antibiotic exposures. So what do we know what happens in the emergency department? I think this is a really nice study that came out of the Netherlands last year, where Kohlberg et al. Looked at 16,000 adults who presented to the emergency department, and they interrogated the antibiotic exposure that they received during this time. They defined antibiotics as either anaerobic or non-anaerobic, where the anaerobic antibiotics were really our piptazo, meropenem, augmentin, clinda, and metro, the really hard-hitting, big anaerobic spectrum antibiotics we're all familiar with. And they found that after adjustment in multivariable models for all other factors, that exposure to anaerobic antibiotics alone was associated with decreased overall survival at 30 days, and interestingly, decreased infection-free survival at 30 days. Similarly, looking at general hospitalized patients, this was an enormous metadata analysis coming from the US, where Baggs et al. Looked at over 500 hospitals and over 12.7 million hospital admissions, and divided the exposure that patients received into high-risk antibiotics versus low risk, where the high risk were those third and fourth generation cephalosporins, quinolones, beta-lactam, beta-lactamase inhibitors, carbapenems, and oral vancomycin and lincosamide. And they have found that after adjustment for all other variables in univariate analysis, that exposure to these high-risk antibiotics, the majority of which are anaerobic, was an independent predictor of subsequent sepsis or septic shock within the following 90 days, and that the cumulative exposure to antibiotics and the number of unique antibiotic classes were also associated significantly with subsequent risk of sepsis. So this idea building that an exposure to antibiotics leads to subsequent downstream effects in a temporal way. Looking in the intensive care setting, Chandaraja group from Michigan did a really nice study looking at 3,000 all-comer ventilated patients in the intensive care. And they found that any anaerobic antibiotic, where anaerobic antibiotics were defined by IDSA criteria, quite broad and inclusive, but very reasonable, high-risk antibiotic exposure, was associated with increased hazard of ventilator-associated pneumonia and ventilator-associated pneumonia death 30 days after ICU admission, decreased infection-free survival, and decreased overall survival at 30 days. And I want to point out, if you ever want to read this study, I think they do a really nice job of trying to address what's probably the biggest challenge in looking at these big retrospective cohorts, which is the potential to confound antibiotics by indication. And so the fact that the reason you're receiving a broad spectrum antibiotic may actually be the reason that you do poorly rather than the antibiotic itself. And they do a really nice analysis looking at all of the microbiological results that these patients receive and determining whether antibiotics were appropriate or not. And then in a subsequent sensitivity analysis demonstrated that all of these observations still persisted. And this group have gone on to publish, hot off the press, a paper that's received a lot of traction on Twitter and in the media in probably the last 2 weeks, as it was released earlier this month. They looked more broadly at the experience in hospital for patients who presented with sepsis in the setting of having been in that period of time of piptazo shortage and switched to cefepime usage. So they had some 7,500 adult patients who they included for 4,500 people who'd received piptazo, and the other 3,000 or so received cefepime. And they first demonstrated that there were no fundamental differences in those cohorts who received piptazo versus cefepime. And then they went on to look at the outcomes of these patients and demonstrated that piptazo was an independent predictor of an absolute mortality increase of 5% at 90 days following admission. That piptazo was associated with 2 or more fewer organ failure days, 1.1 fewer ventilator-free days, and 1.5 fewer vasopressor days, which is pretty stunning. So I think probably the other setting that I'm really interested in from my research perspective, but I think because they're a really unique population where the immune response is absolutely critical to patient outcomes, is what is the potential role for antibiotics to disrupt outcomes in the setting of cancer and the immune compromise. I think this is a really nice systematic review and meta-analysis that actually came out of a group in Sydney, where they looked at antibiotic exposures or multiple cohort studies that interrogated antibiotic exposures and response and overall survival in patients who received immune checkpoint inhibitor therapy. And what they did was they divided these cohort studies, pulled all the patients together just short of 3,000, and divided the antibiotic exposure for the purpose of the analysis into antibiotic exposure that occurred before checkpoint inhibitors, antibiotic exposure that occurred around the time, and so immediately before and immediately after checkpoint inhibitor exposure, and antibiotics that occurred after checkpoint inhibitor initiation. And in their meta-regression, they found that there was a really strong association between antibiotic exposure window, so the time at which you experienced antibiotics, and the subsequent progression-free survival and overall survival of patients. And where the worst association with adverse outcomes was those antibiotics that preceded immune checkpoint inhibitor. That there was really no difference or excess contribution to survival and progression-free survival when you received antibiotics afterwards, which I think is quite nice evidence from this pooled analysis that there really is a temporal association between the antibiotics you receive and subsequent adverse outcomes. Similarly, in the setting of CAR-T, a group from America who pooled their experience with some 300 CAR-T patients from Memorial Sloan Kettering and the University of Pennsylvania, divided their patients into those that had either received in the 30 days prior to CAR-T broad spectrum antibiotics, which were really piptazo, imipenem, or mero, versus not. And they found that any exposure to those antibiotics was associated with reduced overall survival, and in a setting of this subcohort of patients who had non-Hodgkin's lymphoma, associated with an increased risk of immune-related adverse events. And finally, in the setting of allogeneic stem cell transplant, consistently across multiple cohorts, carbapenems, beta-lactam, beta-lactamase inhibitors, so our meropenem, imipenem, and piptazo, during the period of neutropenia, after stem cell infusion, has been consistently associated with acute graft versus host disease and worse overall survival when compared to non-anaerobic antibiotics. And I think really interestingly, there's a study being undertaken by really the pioneers of this research at MSK in New York, where they're randomizing patients during their period of neutropenia to either piptazo and cefepime, and they're serially measuring the microbiome in these patients to demonstrate the influence of that randomization of antibiotic exposure, and measuring microbiome outcomes and patient outcomes in that cohort to see if we can really get to the bottom in a more rigorous sense about what antibiotic we should be using in our cancer patients. And so I think that probably raises the question about, is there a special significance of the role of antibiotics in the setting of the immune compromise? And that comes back to this idea that there's that reciprocal essential interaction between the gut microbiome and the immune system resident in the gut. We know that graft versus host disease, the immune-related adverse events, particularly colitis, that immune checkpoint inhibitors patients experience, as well as the cytokine release syndrome, which is obviously an immune phenomenon in the setting of CAR-T, are inherently immunological and are often occurring at that interface with the gut, with the colitis presentation. And so without there being enough time to really go into that literature today, that's sort of an entirely different cohort. There is increasing evidence to support the importance of this interaction. And so, for example, for immune checkpoint inhibitors, if you were to give germ-free mice, so bacteria-free mice, administration of the microbiome in the setting of a cancer model, so you give mice melanoma or sarcoma or whatever the cancer may be in the setting of having no microbiome, if you gavage them through the mouth, a population of bacteria, you can demonstrate spontaneous anti-tumor activity simply from the presence of gut bugs. If you antibiotic deplete these mice once they've had a microbiota develop, you can actually render immune checkpoint inhibitor therapy when you expose these mice to anti-cancer treatment completely ineffectual. And then when you, again, re-oral gavage those bacteria, you can restore that anti-tumor effect of the immune checkpoint inhibitor. So clearly this has got to be a focus of future research and may end up presenting, really interesting novel opportunities, whether it be with microbiome manipulation or probiotics or dietary interventions, whatever the case may be, to optimize microbiome to improve immunological responses. So I think in summary, I absolutely cannot do a talk that even touches on the microbiome without the cool emoji. Hopefully I've sort of given a sense that there is an interaction between the immune system and the microbiome that is essential, which sort of underpins this idea that, what are the untoward effects of the antibiotics we may use. I think antibiotics, particularly anaerobic spectrum antibiotics, disrupt the microbiome, with a real focus being on our important anaerobic microbiome, and that this is supported by a number of preclinical and clinical models of antibiotic exposure, so challenge studies, and that these clinical cohorts, which are quite rigorous and large, demonstrate that these broad spectrum antibiotics are independent predictors of worse outcomes, and not just the traditional C. diff and the acquisition of MDROs. There are obviously with these studies, I've mentioned it earlier, inherent limitations with the methodologies of doing these retrospective studies, and authors have variably tried to address those potential confounding factors, but it sort of doesn't change the fact that I think that the observations really are consistent, that there is a signal here that warrants further investigation. So I think future directions really need to tease out the effect of those anaerobic commensals in critical illness in the setting of cancer and immune compromise, and it incorporates studies that include both the antibiotic exposure, microbiome sampling, immune phenotyping patients, and then linking that to outcomes. And there is already quite a bit of science to support that as a concept, but that really is material for another talk. And which I'm happy to give one day because I could go on about it forever. But hopefully I've been able to demonstrate that there are reasons to think more broadly about the effects of the antibiotics that we prescribe. So thank you very much. Thanks for the opportunity to talk today.
Oh, thank you so much, Olivia. That was fascinating. And it's really exciting to see that there are going to be some future opportunities to really address the microbiome and improvement of it post-antibiotic use. For anyone who's got questions out there, please type them through, or we can also unmute you. And Kaz has already got her hand up, so please go ahead.
Great talk as always, Liv. It's really a question about what the message is going to be and how to deal with the risk, because of course you've got this challenge where you will have patients presenting to primary care and hospitals with sepsis syndrome that needs to be empirically treated, and clearly even just a short empiric treatment is enough to have an impact. How do we deal with the intended and unintended consequences of this going forward? Because people, we now this is a very strong message, of course. So basically, it's almost like any antibiotics. So what do we do? Like, there needs to be, for the stewards in the online today, what is going to have to happen? Is there going to have to be, you know, immune, you know, microbiome reconstitution post-treatment, or what do we do?
It's a great question. I don't know there's an easy answer. I mean, I think about this a lot actually, and how it affects, you know, my prescribing day to day. I think, you know, I'm pretty upfront with patients now that anytime I recommend an antibiotic, I've mentioned upfront, you know, no good deed goes unpunished. But, you know, there is a good deed in the first instance, and we're using antibiotics because we need to. But I guess there's a lot more literature, which again, I didn't have time to touch on today, but that, you know, we probably can shorten the duration of antibiotics we prescribe. There are, you know, there's a nice paper that came out recently demonstrating that there was no additional benefit with anaerobic spectrum aspiration and hospital-acquired pneumonia exposure. You know, we're increasingly building a body of literature that supports shortening what are perhaps historical dogmas about duration, that there are opportunities where we don't need to target anaerobes, so we can narrow our spectrum. And then I think the broader piece of work are the work that, say, Memorial Sloan Kettering are doing, where they're, you know, rigorously interrogating this in the setting of a randomized study, exposing patients to either an anaerobic piptazo versus cefepime in the setting of neutropenia, again, challenging that traditional dogma where we've, you know, really used tazocin historically. So I think there's a larger body of work that is underway across a number of, you know, stakeholder settings and cohorts to support this concept that shorter is better, narrower is better, and hopefully that will lead to better prescribing over time. But I don't think there's a quick fix.
I did want to pick up on that issue around cefepime and because actually the patient level analysis of cefepime and neutropenia fever was the opposite, that it was actually cefepime that was associated with increased mortality. Can you comment on that? Because it's the complete opposite to what the FDA found, and they looked at it very carefully, and I think the effects still remained.
It's hard. I think it's difficult. Anytime you start looking at influences of the microbiome and using antibiotics as a surrogate, it gets quite murky because the microbiome, is such a dynamic beast, and there are so many other influences, especially in the setting of a cancer patient and an inpatient, that are unable to be taken into account, like diet, other medications, which are, I think, is equal. We underappreciate how significant all those other factors are. So I think it probably is it's an oversimplification to simply think it will be antibiotics making those significant differences. But I think the thing that's nice about the Memorial Sloan Kettering study is they are incorporating a lot of those things into their randomized trial. They're collecting very detailed dietary analysis, and it's in a single center, very detailed drug exposure data. So perhaps we can go away towards that discrepancy between, you know, all these retrospective cohort studies and animal studies which suggest cefepime is better than piptazo, and perhaps some of the older studies that suggest it's otherwise. I don't have a quick answer, I have to say. I think it's complicated.
And I don't think you didn't touch a lot on metabolomics, but obviously that's the other key to this, which people may not realize. It's sort of like the end point of your microbiome. Like, what are the metabolomes that are impacting response to treatment and infection? Did you want to just briefly comment on those?
Yeah, 100%. I think absolutely. Anytime you look at microbiome papers, you know, you're really looking, when you're looking at what bugs are there, it just simply tells you who is present in the sample. And there is a lot more to the story that I've hopefully mentioned, that, you know, bugs are essential for metabolism. They interact with the immune system. And when we simply measure the presence or absence of bugs, we're not analysing any of those things at all. So the metabolome, so the metabolic activity of the gut bacteria, the short-chain fatty acids that they're elaborating may actually be more important than simply who or is not there in the zoo. And similarly, when you look at the host response, that when we're immune phenotyping the host and how the host is responding to those short-chain fatty acids, I think that's really important. So it's oversimplistic simply to just look at the presence of bugs in the gut. You really want to try and have a sense of what gut bugs are there, how they're behaving, looking through the metabolome, and how the host is responding through an immune phenotype to then link that with how a patient does longer term.
And my last question, so for people online, is it expensive to do microbiome-based research? And how hard is it collecting poo samples? Because everyone says, oh, let's do a microbiome, let's do a study on this. Why don't you tell the room exactly what it's like?
Thank you for acknowledging the last couple of years I've spent collecting poo samples in tubes. Interestingly, collecting samples is not that hard. You just have to get a patient on board with collecting it. So, it's a waste product that otherwise would be flushed down the toilet. And it's astonishing actually how aware I was stunned by this, how aware the general population is regarding the importance of the microbiome and how motivated they are to participate in these kind of studies. I have a very low refusal rate, which is really, excuse me, very interesting. That's sort of the easy part in some ways. It costs about $20 to get a collection tube to preserve microbiome features. So that's pretty straightforward. But once you start trying to process and analyze samples through sequencing or metabolomics, then you're sort of getting up into a couple of $100 per sample. And then you have a very large aggregate of metadata, which requires quite sophisticated analysis and manipulation. And that obviously requires a lot of resources as well. So I have increasingly, over my PhD, trying to do some of this work, gained a lot of appreciation and respect for a number of these studies, because it's obviously a huge amount of work.
Thanks, Liv.
Well, thank you so much. I have so many more questions, but I think I might have to pause them for the minute and introduce our next speaker. So thanks, Olivia. Tony, I'd like to welcome Tony online now, and many of you will know him. He's been around with us for 20 years as a pharmacist, I think, and he's a current senior AMS pharmacist at the Children's Hospital at Westmead in Sydney, and an honorary clinical lecturer at the University of Sydney School of Pharmacy. So Tony's here today to share his experience with phage therapy. So over to you, Tony. Thank you.
Thank you. Thank you for the opportunity to speak. Nice chilly morning indeed. I have no conflicts of interest in relation to this talk. And just before I get into it, I just want to acknowledge the shoulders of giants. I sit on with John Ardell's lab, who's really leading all the research and clinical use, along with Aneka Tami, one of the pediatric ID physicians I work with, and a supportive director of pharmacy, Peter Barclay. So the overview of my talk for the next 20 to 30 minutes is just to go quickly over the problem, what phages are as a solution, and what pharmacists really need to know about phages, because what I'm often getting correspondence of is that pharmacists, the pharmacy departments, are often the barriers to accessing phages when you've got a very proactive ID clinician wanting to use it, and they're very complicated ID patient. So this, I'm hoping to fill that gap in for the pharmacists in this virtual room. So the problem, I'll just go over this really quickly. You all know about AMR and the O'Neill report, which is commonly used in grant submissions of the financial impact of AMR. But something I feel like is a bit underdescribed is that our life expectancy is much longer, and we're more reliant on devices, ECMO, prosthetics, and we have much more innovative care with immunotherapy, and therein lies the opportunity for infection. And it's a global issue, as we all know. So what happens when we do get a patient with a multi-drug resistant infection in our hospital is we have to resort to last line antibiotics. And what's arguably one of the most broad spectrum antibiotics on the market in our armamentarium is cefiderocol, which is active against most multi-drug resistant gram-negative infections. And it was marketed and released in 2019. It only took 2 years for cefiderocol resistance to be described in the literature. And this was published in CID with a 58-year-old liver transplant patient with a bloodstream infection, who sadly died 21 days after due to no other antibiotics to give for this patient. So the solution, one of the solutions is phages. So as Mita Sharani is given great commentary on how we've used very militarized terms in the AMS space of the combat AMR and the war against AMR. And I think phages really fit in perfectly with this theme where it truly is the enemy of my enemy is my friend. And phages are the natural enemy to bacteria. So just remember that phages aren't a new thing. They have been around for billions of years, which I'll touch into later. So I firstly want to describe our first case of the successful case of using bacteriophages in a 7-year-old girl who had a car accident in India and had her orthopedic surgery in New Delhi. Came back to Sydney, came to our orthopedic outpatient clinic, and we swabbed her wound and we grew this really scary Pseudomonas. And looking at the antibiogram, you can see it's resistant to all our sexy beta-lactam, beta-lactamase inhibitors, with only limited options with colistin and cefiderocol. And at that time, we threw all the colistin and astrinum at her, debrided and removed the hardware. And after 3 months of quite intensive antimicrobial treatment, the infection looked like it was progressing based on imaging, tracking down to her ankle. And the next option was an amputation, which is quite a significant intervention for someone so young, and really changing the course of her life if we went down that path. And this is her now, and this is well celebrated. You can find this online in the Sydney Morning Herald, where we gave a 2-week course of phage therapy in combination with some antibiotics. So at the moment, at the Sydney Children's Network with Westmead and Renwick, we're accruing quite a decent clinical experience with patients, up to 6 patients now. So our first patient there in 2019 was the one I just described, and we've given it to CF patients with mycobacterial respiratory tract infections, a BMT patient with a nasty multi-resistant pseudomonal infection, and more recently, we've given it for an MRSA and a SUD infection in a young kid with post-orthopedic surgery complications. So what are phages? So phages, bacteriophage, comes from the Greek word to devour or eat. So they're basically bacteria eaters. And like I said before, they're not new. They're nature's own enemy against bacteria, and they're everywhere. They're ubiquitous. If you took 1 ml of seawater, there would be over 10 million phages in that little 1 ml sample. And there's 10 to the power 23 infections per second happening in nature right now. So this was first described by this French-Canadian microbiologist, and the main sources are harvested in the environment or isolated in a lab, specifically for treatment. So here's a nice picture of Ruby Lin, one of the scientists in John Ardell's lab during the COVID pandemic. She was going through sewage trying to harvest some phages. So how do phages work? They act like other viruses, but they strictly, specifically infect the bacterium, spreading the bacterial viral DNA and causing lysis, so cell death. Okay, so just remember that lysis is an important mechanism of action in how phages work, because there's a whole other pathway which I'll talk into a bit later, which we don't want happening. So here is a nice collection of magnified photographs of how phages, how they infect and what they look like. You can see it causes lysis in this cell right here. And I guess what all the pharmacists are always asking about is what's the product information? What does it look like in terms of phages, how it's given, adverse effect profile? And it's most commonly given, certainly within the Australian context, as IV and topical treatment. Usually it's once a day or twice a day, 5 to 30-minute infusion, and or nebulized. And so far, Phage Australia has a decent bank of staph, SUD, and mycobacterial phages, and it's it's described to be very safe to give, with mild transient systemic inflammatory response, so raised CRPs, very short-term adverse effects, and this is supported with a more recent Lancet ID systematic review where they found 9 RCTs showing that the adverse effect profile is less or equal to the control group. Admittedly, most of these studies were low to moderate quality, but there needs to be more research in this space looking at the clinical improvement endpoints. So we know from what's previously published that the toxicity profile is good, and it's costly. So it's only available by compassionate access through Stamp Australia. It's free, but it cost John and his lab about 40K to put a phage together for your patient. So something to be mindful of. Now, if we compare antibiotics to bacteriophages, they're very different pros and cons. So we have an excellent understanding of the PK/PD of antibiotics, but it's well known that there's high risk of resistance. If there's no blood, there's no drug. So there's poor penetration to biofilms and prosthetic material. There's a large range of toxicities, very frequent dosing and long durations, which is very a unique pro of bacteriophages, where it is quite short durations of treatment. What is really important is the lag of access to novel antibiotics. So if we have a septic patient with a very resistant gram-negative bug and we want to give cefiderocol in your rural hospital, the access of it is quite limited because, one, Australia is a small market, it's a SAS drug, it's not TGA approved, so there's lots of admin and logistical barriers to access the antibiotic in a timely fashion. And then we got bacteriophages with lower risk of resistance, and they have excellent activity against biofilm. But with the literature needs a resurgence because it's based on a lot of the literature was based in the pre-antibiotic period, so before penicillins were discovered. Phages were used quite a lot in the Eastern Bloc, but now there's a growing resurgence now, certainly by the Belgians in this space, and there's poor descriptions of the phage kinetics because it's very different to pharmacotherapy PK with a clear absorption, distribution, metabolism, elimination PK curve. So what does that mean to me as a pharmacist? So I think this is really compelling. Certainly, the experience in the UK is there was this great publication from Great Ormond Street, considered one of the best pediatric hospitals in the world, where they had this successful case of giving GMO phages for a CF patient with Microbacterial processus infection, and it was a successful case. And this is the patient here. So this patient transitioned to adult care and actually changed postcodes and was in a different catchment area, NHS area. So went to a different hospital at a later stage of her life. And the hospital there were very unfamiliar with phages. And these were the comments that they were making to the mother, as they were refusing care because they weren't sure of what it is, what phages were, and they doubted its efficacy. And they were making comments that they were shocked that she was still alive. So she sadly passed away in February 2022. And this is, I think, an important anecdote to consider when we talk about patient equity of access of care. When you've got a patient who's admitted to the Alfred, which is a stamp site, and then a patient who's admitted to another hospital in Victoria, which have not registered as an investigative site for phages, and haven't assessed their organisational readiness to give these type of what we consider investigational treatments. And this was followed by a parliamentary inquiry in the UK, to which John O'Dell was actually asked to speak to, an Aussie speaking in a UK Congress. So what do we need to know with the STAMP study? So the STAMP is a protocol multi-site study, which quite a lot of the Australian hospitals have already registered investigator sites for, and with full HREC approval, and the pathway of access is via compassionate access through the SAS scheme. And when you identify a patient, you send your culture, your micro, your SUD to Phage Australia, and we check for susceptibilities to that phage, and we grow the phage to that specific bug. And then you've got to sort out your drug committee governance approvals. So we isolate the host bacterium, and we use that host bacterium to grow our phages for that patient specifically. So then after that stage, John and his lab will send you all this documentation for your drug committee for governance approval. And this will be the proof of sterility, the concentration and stability, so kind of GMP standards, trying to tick a few boxes. But what his paperwork will also show that the phages that he's made for your patient is free of lysogenic markers. So you know how I mentioned before that a lysis is great, but lysogenic is bad. So causing lysis causes cell death and an effective kill of the bacterium. But then there is this other pathway, this lysogenic cycle, where the phage integrates its DNA into the bacterium, and the bacterium doesn't die or lies, and it continues spreading that bacterium. So why is that bad? Because it's clearly a less effective kill. There is unpredictable effects of what the further downstream effects are of that integration of phage DNA, the prophage. There's potential for resistance, and there's horizontal gene transfer. Okay, so you need to check as a drug committee panel member. You would always check, is this free of lysogenic markers? And also because you are using bacteria to grow those phages, you need to make sure it's free of antimicrobial resistance and virulence genes left over. And there are no endotoxins, or no, sorry, I corrected. There's endotoxins that are less than the FDA agreed level, which is described to be less than 5 endotoxin units per kg per dose. And that's what causes the adverse effects, the fever, chills, CRSD type of response. So the fifth question is it a GMO phage? Now, this is an important question because what we need to consider with, so you got your natural phages, which occur in existence in the environment, but you've got GMO phages. So we need to control the ecological impact of potentially releasing GMO phages into the environment. And that's where there's a lot of containment required if it is a GMO phage. So there's more work required, and you need to really assess whether or not your facility can handle GMO phages, which is almost like a gene therapy level occupational exposure and handling, where they need to be made up in a PC2 lab. So there's the GMO phages on the right. This is all described in one of our policies in New South Wales Health, which you can look into a bit later. But when they're non-GMO phages, they can be sent to the ward, made up by the nurses and given because they're naturally occurring. It doesn't matter if it spills on the floor. Whereas it's GMO phage, it needs to be contained. This is me taking selfies of making up some GMO phages in our PC2 lab. And what I might move on to quickly next is what I feel like is a lot of unknowns about phage therapy is the dynamics, the phage dynamics. And there's this excellent narrative review written by a close friend of mine, Indy, and Amine, our RD physician. And you might be familiar with these type of diagrams with antibiotic replacing the phage. cartoon. But there are really poor descriptions of what the dynamics are of phage to the patient and to the bacteria. But one must be mindful in the background, in all patients who are given phage treatment, there is always adjunctive antibiotics given. So you've got to consider if there's actual antagonism or synergy possibly happening with the phages. And this is a massive gap in research that needs to be filled, whether or not there are preferred antibiotics to be given with phages, whether or not certain combinations improve its biofilm activity. There's this really great publication by Armine and John. So this was actually looking at the kinetics in a phage patient of that 7-year-old who had that car accident with the SUD osteomyelitis, chronic osteomyelitis. We looked at the kinetics. So this is a really busy slide, so just bear with me as I explain. So we've measured quantitatively the bacterial genome copies, so the quantitative counts of SUD, CRP as a surrogate marker of a SIRS adverse effect profile. And then we've got quantitative copies of the phage, which is the PASA 16. So they give really weird names of phages. Like one of the phages we gave was called Muddy. I don't know how they come up with it, but the phage that they gave was called PASA 16. And you can see the days of treatment with phage doses given as gray arrows. And just pay particular attention to the orange curve where there is high increasing amounts of SUD copies. And then we give a few days of phages, and then that increases in the blue line to quite a high exponential level. And there is, as a lag, behind the SUD, because that's clearly the target of infection. And then there's a massive drop of SUD with a laggy drop of phage. And this is what's been described in, I guess, biochemistry, is a predator-prey relationship where there's large amounts of prey and you introduce a predator, there is a drop. So this is when more descriptions of this dynamics need to be made, and looking at the effect of the adjunctive antibiotics given in the background. So that was quite a brief talk on phages, and hopefully I've given an orientation to the clinicians in the room about this. But this is a very popular meme that's been spread around, or as actually I think it's actually considered the most popular meme ever used. And this is I guess the thing that we'll be coming up against, is that instead of using novel antibiotics that will have antimicrobial resistance to that new drug, we might have to consider other novel treatments like phages. I'm happy to take any questions.
Thank you, Tony. Yes, please send your questions through or type them, and we can also unmute you as well. Yeah, that was just...
I'll try get in before Kaz. Great talk, Tony. Just wanted to clarify the stamp protocol, the screenshot you had, that was for pediatric use only. Is that correct?
No, pediatrics and adult. Yeah.
And adult. Cool.
Yeah.
Cool,
Yeah.
cool. And then you said that it cost the Ardell lab around $40,000 to make the phage. So essentially they would get compassionate act, like the pharmacist or the other, for that patient, you would get compassionate access for the phage. But like you said, it's not just like here's a tablet, take it. Like you have to then design the phage around the patient's like the bug and the profile. That's where that $40,000 comes into play?
Yeah, and along with all that, the GMP standards of sterility,
Yeah, the
concentration,
quality.
endotoxin levels, yeah, all that quality assurance documentation, that's actually the bulk of the cost, yeah.
Yeah. And then so like the hospital, like not your hospital, but the hospital with the sick patient, they fund that, or is there
No. So that's
restriction on
that's
that,
free. So
or
it's
that's all part
compassionate.
of it?
Yeah. So it's free to the stamp site, but I was just describing how much it actually costs in the
Oh,
background
yeah,
for the
yeah.
lab to
Yeah,
be,
no. Yeah.
because it can be quite annoying when sometimes when people get something for free and they don't really realize
They
the
don't
cost.
appreciate it.
You guys need to appreciate that you guys got approved to use this phage out of a long list of patients, and you're creating all these barriers to use it. So yeah, that's what kind of why I put into
Yeah,
that doc.
yeah. No, that's really insightful for everyone listening too. No, thank you.
Yeah.
Sorry, Kaz, what's your question?
No, it's such an interesting area, isn't it? And it's really interesting to see where it will progress in terms of scalability, Tony. And I agree with you. I think the amount of work that goes into the product approval as a medical as a medication and approval at all the activities, the untold costs. We know the cost of developing an antibiotic goes into the billions with drug companies, and one can probably imagine that scaling phage therapies as some sort of standard that's readily accessible is going to be similar. And so it's very impressive when organisations like yours have been doing the work that you're doing, you know, that early development work. So you must have had discussions around the lunch table around how feasible the scalability of it all is. I'm really interested in your thoughts.
Yeah. So with brief conversations with John, he's trying to deliver this service kind of like a transplant type of model of care, because phages, yeah, it's really difficult to get funding for, even through MRFF grants. He's gone through hedge companies, and they're all looking at return of investments, which is not the business model you can have with phages. So that's why I think his approach with having it like similar to a state liver transplant service would probably work, but yeah, it is really costly. And I guess one of the great things about having a phage bank that's accessible nationwide is that you can try to imagine having an antibiotic, so a deck of cards of antibiotics you could flick through, and pick the one that's for your patient for that bug, and rather than having to wait for novel antibiotics to go through research and development and FDA, TGA approval, which takes decades to occur. So yeah, it is a difficult space with the scalability, but I think the direction that John's going towards seems like a clever one. Yeah.
One of the important things for all the highly specialized treatments is being able to triage who gets it when. Like, when do you pull the trigger? So when would you pull the trigger to go to phage?
That's a great point. It's certainly not used, first line empirical treatment. It's last line treatment. It's often with retained prosthetic material where you've thrown a lot of antibiotics out already with a horrible resistance profile. So they're the type of factors that you consider that would make a good candidate for a phage patient. Yeah. Where we know that there's a biofilm there, and you're just not going to get any antibiotic to the site of infection.
Yeah, thanks, Tony. Fascinating.
Very important therapy for the last line options, as a last line option. So it's fantastic to see. And also, just out of curiosity, what's the general shelf life of phage therapy once it's provided to an organisation or produced?
1 month. Yeah. So they're quite short. Yeah.
Okay, so that makes it hard to have a banking system
Yeah.
set up as well, if you're putting in all that cost as well. So I can see some of those limitations there.
Mm.
All right, and I haven't seen any other questions online. So what I might do is wrap up this session. So thank you so much for your talk, Tony. I think that's just so fascinating, and we've got a lot to learn with the phage therapy, but it sounds very promising at this point in time. And Olivia, thank you so much for your presentation as well. We might take you up on your offer for those other related topics at some time in the future, because we've got lots more questions, I'm sure, and also increasing evidence as you progress on your PhD. So thank you very much. Courtney wants to say something as well, so I'll pass on to her.
Thanks, Ngan-Ha. Just wanting to say the next month's topic is aged care. So watch this space and looking forward to seeing you all online for that. And also to save the date in November for Antibiotic Awareness Week, we'll be hosting an in-person event on the Friday, the 22nd of November. So more information will be disseminated through our NCAS and MAPS communications about that. And yeah, have a lovely day, everyone. See you all around.
Enjoy your day. Stay warm.
Thanks, Tony, Liv, Ngan-Ha, Court.
Thank you.